Welcome back everyone to MMBS111, Introductory Microbiology. This mini lecture is going to cover section 6.1, Microbial Nutrition, and we have quite a bit to talk about related to how microbes get their nutrients. There are 5 learning objectives for this mini lecture. The first, be able to list the essential nutrients of a bacterial cell, and that includes macro and micronutrients, as well as the 6 major elements. Be able to define the following terms: photoautotrope, chemoautotrope, photoheterotroph, chemoheterotroph, saprobe, and parasite. Explain diffusion versus osmosis, be able to distinguish the two. Identify the effects of isotonic, hypotonic, and hypertonic conditions on a cell. And finally, explain passive transport, both simple and facilitated diffusion, as well as active transport, and name the 2 types of endocytosis. So let's start with the nutrients. What do microorganisms need? So all living things need some essential elements, and we call those the macronutrients. They're macronutrients because they are found in large amounts within living things. There are 6 elements that make up most organic compounds. The first is carbon, and carbon is essential to all organic molecules. It's not organic if it doesn't contain carbon. Then we also have hydrogen. Hydrogen is involved in a number of things, primarily bonding of things like DNA strands, proteins to create that secondary structure. They are also involved in maintaining pH within fluids, and they are an important component of cellular respiration and energy production. Oxygen is also another one of those essential elements, and they are involved in pretty much every organic molecule. Carbohydrates, lipids, nucleic acids, and proteins, we're going to find oxygen within those compounds. Nitrogen is essential in nucleic acids. It's also an important component of amino acids and proteins. Phosphate, also an important component of nucleic acids. That backbone of DNA and RNA is composed of phosphates and sugars, ATP. Without the phosphate, we don't have those high energy bonds. And then sulfur is also an element that's important in many amino acids. It's also a component in a number of vitamins. Then we have some other nutrients that are important, but just found in smaller amounts within living things. Sodium is an important element in cellular transport. It helps to create gradients without the cell membrane to move components across the membrane. Calcium is another essential element. It's important in the cell wall structure of a number of microorganisms, and it is a very important component of endospores. Magnesium is found in chlorophyll. It's also a component of cell membranes and ribosomes. Iron is an essential element in cytochrome proteins. These are proteins that are colored, they're pigmented, and they're involved in cellular respiration. And then zinc is also an element that's involved in regulating eukaryotic genetics. So all of these elements are important. They're just found in much smaller amounts. So when we're talking about microbial nutrition, there are 2 ways that organisms can get their nutrients. They can either get it from an inorganic source or an organic source. So when we're talking about inorganic nutrients, these are things that have never been living. They are elements of the Earth's crust, water, the atmosphere. Carbon dioxide is considered an inorganic nutrient. So these are atoms and simple molecules that contain combinations of atoms other than carbon and hydrogen. Organic nutrients contain carbon and hydrogen atoms, and these molecules are the products of living things. Things like methane. Methane is a simple byproduct of organic molecules. We also have these other large polymers that we've already talked about, like carbohydrates, lipids, proteins, nucleic acids. These are all organic compounds and would be considered organic nutrients if consumed by another organism. So this chart here helps to break down how we classify microorganisms based on their nutrition. And the first thing that we look at is the type of nutrient. Where do they get their carbon? Do they get their carbon from an inorganic source or from an organic source? If an organism gets its carbon inorganically, like carbon dioxide, this organism is considered an autotroph, which literally translates to self-feeders. They can feed themselves. They make their own food. If an organism gets its carbon from a living or a once living source, we call these heterotrophs, which literally translates to other feeders. They rely on others for food, other living things. Then we can break down these two sources of carbon into smaller groups depending on how the organism gets its energy. If an organism is an autotroph, it can get its energy a couple of ways. One way is by trapping light to get its energy, and we call these organisms photoautotrophs. Photo means life, or not life, light, sorry. So these are all organisms that photosynthesize, photosynthetic bacteria, algae, the cyanobacteria. Any organism that is able to use an inorganic carbon source and light for energy is considered a photoautotroph. Another way that an autotroph can get its energy is by breaking down chemicals. When we break the bonds of chemicals, like those found in ATP, we can release a lot of energy depending on the chemical. So an organism that uses an inorganic carbon source but must break down chemicals to get its energy is called a chemoautotroph. And there are two different types of chemoautotrophs. There are the chemoorganic autotrophs that use organic chemicals to get their energy, like ATP. And then there are the lithoautotrophs, which use inorganic chemicals to get their energy. Litho means rock. So these organisms are going to use inorganic sources both for their chemical energy as well as their carbon source. Archaea are typically found in this group of organisms. So now let's look at the organisms that rely on organic carbon as their carbon source, are heterotrophs. We can also have heterotrophs that are able to trap light for energy, and these are called the photoheterotrophs. They use light but rely on organic carbon as their carbon source. So some examples are the purple and green non-sulfur bacteria. And then we also have chemoheterotrophs that use organic chemicals for their energy source. These are organisms that use ATP or similar molecules for their energy. when we're talking about microbes, then we can break down the chemoheterotrophs into some smaller groups depending on sort of how they get these nutrients and energy. So we have the saprobes. Saprobes are organisms like the fungi and some bacteria that are involved in decomposition, and they rely on dead animal and plant material for their food. Then we have the parasites, and parasites are organisms that live in or on another organism to get their food. A large number of bacteria, our helminths, some protists are going to be categorized in this group with the parasites. Okay? So how do these organisms get these nutrients? Well, they have to transport them into the cell some way. And there are a couple of ways that they can do that, either passively without the use of energy or actively. And in this way, they have to spend some energy in order to get these nutrients that they need. In passive transport, molecules are moving in the natural flow of the concentration gradient. We call this diffusion. If you have a glass of water and you put some food coloring in it and you don't stir it or do anything, eventually the food coloring spreads throughout the glass of water, and over time the whole thing is equally the same shade of blue. This is diffusion. Diffusion is the movement of molecules from high concentration to low concentration. And this is what passive transport does. It relies on the principles of diffusion to move molecules across membranes. Simple diffusion occurs generally with small uncharged particles that can easily pass across the plasma membrane. And these molecules naturally want to move from the area where there is a high concentration to an area where there is a lower concentration. And it works very similar if you think about yourself. If you are in a very crowded room or crowded hallway, you don't have a lot of personal space, most people want to move themselves to a place where there is more room, more space. And this is what molecules do. They naturally want to move from the area where there's a high concentration to the area where there is lower concentration. Some molecules are either charged and don't naturally flow across the membrane, or they are too large and they just need some help to get across the membrane. So we have to use a protein within the plasma membrane in order to move those molecules. And this is facilitated diffusion. So facilitated, they need some help, or a facilitator to help them get across the membrane. Active transport moves molecules in the opposite direction, which is why it requires energy, because molecules don't naturally want to move in this way. So we're going to move molecules from an area where there is a low concentration to an area where there is a higher concentration. And we're going to have to spend a little bit of energy and require the use of some kind of protein in the membrane in order to move those molecules in that way. Another form of transport is osmosis. And osmosis is similar to diffusion, except it only involves water. Osmosis is the diffusion of water across a semi-permeable membrane, so a membrane like the cell membrane, which is semi-permeable. It is a form of passive transport, so no energy is required to move the water, but the water is going to move to balance the concentrations of solute. So if you look at the image on the right, we have a container. It's separated with a semi-permeable membrane. On one side of the membrane, we have a concentrated sugar solution. On the other side of the membrane, we have a dilute sugar solution. So there's a high concentration of water on the right-hand side compared to the left-hand side. So the water is going to want to move from where it has a higher concentration to the area where there's a low concentration of water, but a high concentration of solute. Solute is dissolved particles. And you can see, as we follow the arrow to the far right, the water molecules are able to pass through the membrane, but not the sugar molecules. Since the water can move, it can help to balance and even out the concentration of the sugar solution. So water does interesting things to cells when concentrations are different. So we're going to look at some different concentrations here and see what happens to cells when they are placed in these concentrations. So an isotonic solution is a solution where water moves equally into and out of the cell. So the rate of diffusion across the cell membrane is the same. Water is leaving the cell at the same rate that water is entering the cell. So the cell maintains its normal shape. In a hypotonic solution, the net diffusion of water is into the cell. This does not mean that water isn't leaving the cell. What it means is that more water is entering the cell than is leaving the cell. This swells the protoplast, that internal structure of the bacterial cell. The plasma membrane gets pushed very tightly up against the cell wall, but because most bacteria possess a cell wall, the cell wall helps to prevent lysis, so the cell does not rupture, it just swells. In cells without a cell wall, then we have a problem. And this happens, can happen in eukaryotic cells without cell walls. If you think about your red blood cells, as water flows into the cell, the cell continues to swell, and then it will eventually burst because that membrane can't continue to expand indefinitely. The opposite happens in a hypertonic solution. Here, water is diffusing out of the cell at a greater rate than water is entering the cell. And this causes the plasma membrane to move away from the cell wall. The internal protoplast of that bacterial cell shrinks in, and the cell shrinks down. We call this plasmolysis. In eukaryotic cells, the cell continues to shrink, it becomes distorted, kind of, I don't know how to describe, looks kind of like an amoeba almost, that has a very irregular shape. One way that I use to help me to remember what happens in each of these situations. Hypertonic contains an E, hyper, and an E is kind of a shrunken O. So if you think about that E as a shrunken O, in a hypertonic solution, cells shrink. Hypotonic has an O, and an O is very round and swollen. So in a hypotonic solution, cells swell. That's one way I remember it. So the last way that organisms are going to get nutrients in and out of the cell is through endo and exocytosis. And endocytosis literally means cell eating. Substances, especially very large solid substances, are going to be carried into the cell through the vesicle, as you can see in the image on the left. This process does require that the cells spend energy in order to get these molecules within the cell. Phagocytosis is a process of engulfing very large particles, and this is how our macrophages within our body remove things like bacteria, dead and damaged cells. They literally engulf that whole thing within the cell. It gets surrounded in a vesicle, and then structures like lysosomes break down the stuff that's inside. Pinocytosis is engulfing fluid particles. So this is like cell drinking, where the cell is going to engulf particles like oils and other fluid materials in the same manner in a small vesicle and then bring it into the cell. Cells can also get rid of wastes in this same way. So exocytosis, instead of bring it in, like endo is to bring in, exo we're moving it to the outside. So the vesicle is going to be created around the waste particles. That vesicle will attach to the cell membrane, and then an opening will be created in the vesicle, and the particles will be pushed to the outside. So these are all of the ways that microorganisms are going to get their nutrients and bring those nutrients within the cell. So that's the end of mini lecture 6.1. Make sure you go through these questions, double check that you can answer them correctly and that you have met the objectives in the beginning of the lecture. And hopefully this video was helpful.